Hello everyone, this is Abhinav Chatterjee. I'm currently doing my Masters in Medical Biotechnology at UIC College of Medicine, Rockford. Today I'm going to talk about my project titled Potential Role of Anti-HMGB1 IgM in Sexual Dimorphism in Type 1 Diabetes Mellitus in NOD mice. This research project is approved by the University of Illinois College of Medicine at Rockford Biological Resource Committee. The authors declared that we have no financial or competing interests. One of the subjects of discussion for today's presentation is sex dimorphism. Sex dimorphism in disease is a fundamental phenomenon of the human physiology. Figure A on our left depicts the prevalence of sexual dimorphism seen in different diseases. This is particularly true for autoimmune disorders as well, since in most of the autoimmune disorders, the prevalence of sexual dimorphism is quite significant. As we can see, the incidence of the disease is more in females than in compared to males. For example, in systemic lupus over here, the ratio of the incidence in between females and males is 9:1. Moving on from sexual dimorphism, one of the more important characteristics of autoimmune disorders is the association of inflammation with it. As shown in figure B, different tissues of the body are affected by autoimmune attack. This attack is mainly driven by inflammation. We are, we are going to discuss more about inflammation in our next slides. Chronic inflammation, as we talked about in our earlier slides, is one of the main features of autoimmunity. It is mainly driven by a molecule called HMGB1, which is a pro-inflammatory molecule. It promotes its inflammation through the TLR4 and RAGE receptors, which in turn activate the MAPK, NF-kappa B, and P53 pathways. These pathways upregulate the pro-inflammatory cytokines, which are responsible for the autoimmune attack on different tissues of our body. Our lab has recently found out an antibody against a specific epitope of this molecule, which is endogenously present in each and every person. This antibody is called neutralizing anti-HMGB1 IgM autoantibody or anti-HMW4 IgM. We refer to this antibody as anti-HMW4 IgM because it neutralizes HMGB1 through the HMW4 epitope. Interestingly, this specific IgM also shows male bias in its level of presence in human beings, as you can see in figure B over here. This figure shows that the level of IgM is higher in males as compared to in females. Therefore, we suspect that the lower level of this IgM in females might explain their high incidence in autoimmune disorders. My project is to assess whether this is indeed the case. In order to do that, we have used the NOD mouse model of autoimmune diabetes as my main research model. As you can see in figure A over here, which is a figure presented by Jackson's laboratory, in the course of 30 weeks, about 92% of the females get diabetic in comparison to 50% of the males. This shows a clear indication of sex dimorphism in the incidence of disease in both the genders, as well as the fact that only females develop autoimmune diabetes at a faster rate, whereas the males are relatively protected. Interestingly, the high incidence of diabetes in the females negatively correlates with the low levels of IgM present in the blood. Figure B over here shows the anti-IgM levels in a 12-week-old male and female NOD mice. As we can clearly see from the diagram that the level of the IgM in the males are higher than the females. These observations have led me to ask the following set of questions, which I'll talk about in my next slides. Question number 1 is that does the level of anti-HMW4 IgM in pre-diabetic male and female mice negatively correlate with their blood glucose level? To answer the following question, I'm going to discuss the experimental protocol given by the diagrammatic representation below. In the first step, we needed to check their blood glucose levels for each of these mice every week. For this, we collected their fasting and their non-fasting blood glucose levels. The second step was blood collection. This was done via tail snip as we collected the blood with the capillary tube. This was done in order to process their plasma to obtain their sera, because the sera contains the IgM levels. Finally, with the sera, we needed to estimate the level of anti-HMW4 IgM present in them. This was done via indirect ELISA. This particular slide shows the results of the experiment conducted above. As we can see, the higher levels of anti-HMW4 IgM correlates with the low level of glucose, particularly for females, irrespective of whether it was fasting glucose levels or non-fasting glucose levels. However, the correlation for the males was not that significant. Therefore, we combined both the males and females in one data, which you can see in figure C over here, and we see that the correlation constant we obtained was significant. Coming to our second question, can NOD females be protected from diabetes if their anti-HMW4 IgM is raised? For this experiment, we divided female NOD mice into three groups. The first one was non-treated, which were not injected with anything. The second one was the immunized mice, which were injected with HMW4 specific peptide and MPLA acting as an adjuvant. The third group was control group, which was only injected with MPLA. All these mice were injected intraperitoneally as shown in figure A over here. These injections led to the rise in anti-HMW4 IgM levels, particularly in the immunized groups, as we can see in figure B over here. These levels kept on rising from week 6 to week 8 till week 8 had their levels raised up to 10 folds. After this, with the booster dose at week 8, the levels kept rising till week 18 till the level was up to 300 micrograms per ml. Interestingly, after week 18, the levels started to decay. All the data obtained were checked for their significance via multiple t-tests. This slide shows the onset times for the development of type 1 diabetes for the respective group of mice, namely the non-treated, MPLA-treated and the immunized mice. As we can see in the diagram below, that immunization, that is the raising their levels of IgM, led to delay in the onset of diabetes, particularly for the immunized mice. The immunized mice had their first onset at 20 weeks. This is in stark contrast to the MPLA and the non-treated, which had their first incidences at 16 weeks. Interestingly, the total number of mice which got diabetic at the end of study was also low for the immunized mice. Only 30% of the mice were diabetic for the immunized group compared to 60% of the mice which got diabetic for NPLA and the non-treated group. The significance of the difference was shown statistically by log rank test. Therefore, we can conclude that raising the anti-HMW4 IgM in not females protects them partially from overt diabetes. This conclusion brings me to my last and final question. Can NOD males be unprotected from diabetes if their anti-HMW4 IgM is lowered? Lowering the levels of anti-HMW4 IgM was achieved by injecting these mice with a DOSC complex. This complex depletes the HMW4 specific B cells which are responsible for producing the IgM. The assembly for DOSC complex is shown in figure A over here. This is how we form the HMW4 tetramer, and after attaching the IgG2A monoclonal antibody, we form the DOSC complex. On injecting this DOSC complex, it caused ADCC, that is antibody dependent cell cytotoxicity in these mouse, which led to the depletion of HMW4 specific B cells in the peritoneal cavity, which in turn led to the depletion of the HMW4 IgM levels in their blood. This is clearly shown in figure B, which shows how after the DOSC treatment, it led to the depletion of about 90% of HMW4 specific B cells from the peritoneal cavity of the mice. For the next step, we divided these NOD mice into three groups, that is the non-treated females, which were not treated at all, the DOSC treated males, which were treated with the DOSC complex, and the mock treated males, which were treated with tetramer-less DOSC complex. Their IgMs were monitored from week 2 up till week 26. Figure C over here shows how the DOSC complex led to the lowering of their IgM levels in the DOSC group. As we can see in the diagram below, the green over here shows the levels of IgM for the DOSC. It is lower than both the mock males and the non-treated females. This slide shows the onset times for the development of type 1 diabetes for the respective group of mice, namely the non-treated females, the DOSC males, and the mock-treated males. As expected, the incidence for the non-treated females were early. In between the mock and the DOSC treated groups, although the incidence for the mock treated begin early, we can see that at the end of the 26 weeks, the total number of mice which become diabetic is still higher for the DOSC males, which is around 30%. We have also seen that apart from the 30% of the mice which were already diabetic, there was an extra 20% which remained hyperglycemic for over 2 consecutive readings. This led to a conclusion that reducing the anti-HMW4 IgM levels in NOD males renders them hyperglycemic. The study is still ongoing, so we could only present our data with 90% confidence limits. Finally, I would like to summarize my results over here. The level of anti-HMW4 IgM is reversely correlated with the onset of diabetes. Immunization by raising the anti-HMW4 IgM in female mice reduces their diabetes incidence. And finally, depleting the anti-HMW4 IgM producing B cells in male mice makes them hyperglycemic. From the following set of results, we can conclude that based on the data thus far, anti-HMW4 IgM does appear to play a role in sex dimorphism in diabetes in NOD mice. We have finally come to the end of our presentation, and I would like to take this opportunity to thank whoever has been involved in this project so far, especially my mentors Dr. Gojing Zhang and Dr. Oshang Chen. I would also like to thank Sneha, without whom this project would have been impossible to conduct. I would take this opportunity also to thank Nehal Priya, Dr. Vishal Khatri, Eniola Birch, Mrunali, Nithila and Aishwarya, who have helped me throughout this project. I am also indebted to Jessica and Valentina from the animal facility, who continuously helped me regarding all the problems in with my mice. Finally, I would like to thank Khalifa Suwa and everyone else in the MBT who helped me throughout this project. Thank you.